Today we have actually an easy task because all of you have created different environments or are working in different settings. So first of all, for those of us who are working through the genomics cloud, there are some folks who asked for the access. So you will see the workshop main script. You should open that. And basically, if you did it, you are already here. So, and everything works. The second thing, and I will share my screen now to show how does it look. If you work with your own RStudio, and I guess you can see my screen right now. So if you work with your RStudio, what you should do, so let's imagine you cannot see this because it's not important now. In the lower right corner, there is the files section. And that's very important that you can see it. So the very first thing, and I will ask you to repeat it right now, so we can start this point. You should press File, right? And after you did it, please select the new project. So if you will create a new project, that's how we can actually work with the settings I provided before. So you create a new project, and it will ask you where to create it, and you type existing directory. And actually, then you can point to this directory to which you downloaded the data sets. So the data sets should be there, and you should just type open and create a project. So I think it will work for all of you right now because there is nothing happened wrong. If not, just let me know about that. So what is also important that you should open the script which we'll use today because we'll copy paste a lot from the script. So just for your convenience, I will send the link again to Zoom. But first I will add okay, now I see the chat. So here is the link. Just keep it open because we will not upload this, but we will use quite a lot of this code and copy paste. So what is important that in the file section to the right below, you see this approach file, which is .roj. So this is your project file in the same folder with the datasets folder. And if you open datasets, you will see the hepatocellular carcinoma folder here and health folders here. If it works like that, then everything works fine. The second option, the second step we should all do if you work with the RStudio on your own laptop is to type, under the file button, there is a small file with the plus button. You should press it and type R markdown, the third option. Okay, so I will just do something with my hand because I see that I raised the hand, but I obviously don't want. All right, so we should create the R notebook because this will be our working environment, and I am trying to just explain it slowly because we have people who work with RStudio the first time. You open the R notebook, and it immediately opens such kind of window. So you can just select and delete everything below the upper part, the upper chunk, because you don't need that, and keep open the link which I sent to you because we will copy paste the files from there. So if everything worked for you, the first thing you can do, you can just copy the libraries we import, and if you're a professional user, you can already import the libraries. But since I think many of you are working with RStudio for the first time, first we will do some R magic, so you will understand how does it work. So the specifics of the RStudio markdown document is that we have the code chunks, so to speak. If you will press Ctrl Alt Alt I, it will appear in this, like it will appear like here. I will try to zoom in so you can see better.
If you're a Mac user, it's Ctrl Option I, so it's different.
Or actually, I will just go to the cloud and show it there because here I actually can zoom in. See it better. So for Windows, it's Ctrl Alt I, and for Mac, please do it like a minute ago. So RStudio should be yes. Another important thing, we have two breakout rooms. So if you feel that something is super wrong for you or doesn't work, just ask in chat maybe or unmute yourself, and one of our team members will go in the separate room with you and find a solution. So create a new chunk, and here we will practice a little bit with assigning values to to variables. So for example, if I want to make some variable which will be equal to or any number, I can do like that, like a, the symbol 2. So to create the symbol, you can actually on Windows laptops you can at least press Alt and minus. So Alt and minus create is a shortcut for this specific symbol. Oops. Yes, like this. So to execute some code, you should stand to the on this file and press Ctrl Enter on Windows, or probably Command Enter on Mac. I would think so. And when I do so, I see the console output below. So you see that something happened. I actually assigned 2 to the variable a. And now, if I would just use another line and just do something like this, print a, the output on the console will be 2, which makes sense because we just assign it to 2. Does it? Okay. Is there anybody who didn't reduce it, or did it work for everybody? Because if not, it's better to say right now so we can figure out what's wrong. Okay, one person disconnected. Now she's letting us. Okay. So, okay, fine. Tatiana, did it work for you?
Yes.
Okay, perfect. So I just check in with people who work with RStudio the first time. Now we'll make a little bit more advanced magic. So, some people very inconvenient. Why should we use this symbol, right, to assign something? Actually, you shouldn't. So technically, hypothetically, you can do something like this. So I could assign something to variable b. However, within this course, it's totally illegal, and I would not recommend you to do so unless you have very good reason for that. So the standard assignment value, assignment symbol, is actually to the left. So something like this. Now we will do something more interesting, still basic. So we'll assign 2 values, like x equals 5, y equals 10, and we'll print the sum of these numbers. So first we Control Enter both of these lines, and then Control Enter to the print. And in the output, you will see that 5 plus 10 equals 15. So basically, this very basic examples we reproduce because it's still important for understanding why the code from the rest of the workshop works for us. So sorry if it's boring if you're a proficient user, and all the rest, I think it will be quite helpful. So so we don't use something equals something like that, because in R it has different meaning, and we will sometimes use it inside the functions, but still it's not like in Python, for example. It's just specifics of R. That's the best explanation I can give right now. So what is really important for everything we will do is functions. So for example, if you create a function named my function, and we will assign a function for that. So this is how the syntaxes works. Okay, so we had x equals 5, y equals 10. And we will Control Enter to that. So now how the syntaxes for functions works. I do this figure brackets and type something here. And the second figure brackets actually closes the function. And then I can call the function with the name and try to do something. So for example, if we create a myCool function, which will, for example, create a small temporary variable, which will sum x plus y, but also will add 10. Sounds good.
Sasha,
and we'll ask, yes?
I'm sorry, few participants are in waiting room.
Okay, who is waiting?
It's written that 2 people.
No, who should I add? Because I see a lot of people.
I see, so it's like Igor Stepanov and Isha Goel.
Okay. No, the second person is not signed with this name, unfortunately. I don't see. And okay, so in the end, it's a good idea to ask the function to return us something. And we ask to return this a. Now we will Control Enter on the last line of this function. So R knows we will call that. And now we will actually try to call the function. So we will do my function. But instead of x and y, let's try 15 and 25. Control Enter, and we'll see what happens. Oh, okay. So I did something wrong. It's extremely stupid if I will be mistaken with some syntaxes right now. Yes. And apparently, somehow I fucked up the syntaxes, which is funny enough. Correctly though.
I think it's function without a S. Uh, when my goal function function without a S.
Okay, that's very stupid mistake to make, of course. So it's not functions but a function, and then you call this my goal function, and it should add 15 to 25, which will be 40, and add extra 10. So we will do this, and apparently 40 plus 10 will be 50. Okay, it works. Now thanks a lot for this direction. So now we'll make something more funny. So so this is actually the very basis of any library we create, or sorry, we import. So why we import the Seurat, why we import any other libraries, because they contain the functions, which can be such simple or can contain hundreds of code lines. And well, that's the reason why we do this, because of course we can do everything without any libraries, but still we need to write thousands of the lines of the code, which is not always an optimal solution. So after we apply this function, let's imagine we have not two variables, so we don't want just to summarize 15 and 25, but we have so we'll create by numbers, and we create this magic vector of numbers. So if you want to make at least the vector of numbers, you should add this C and then open the bracket. So for example, 5, 10, 15, 20. And my other numbers, it will be a second vector, and it will be, I don't know, 7, 13, 26, something. And control enter, so our knows this variable. So what we do next, we will try to apply the function which is created for two vectors, right? So to lists of variables. So what we will do, we will use a function which is named apply. So we'll apply my foo function, and then it asks us to provide two vectors. So I will provide as vector one my numbers, and then my other vectors. Okay, let's see what happened. Of course, because they should be actually equal. There is some workaround of that, but doesn't matter for us today. And we print B. And apparently we have the output, which is just a summary of different sum of different numbers from our vectors. So it's important because sometimes functions are applied not just to one value, but for example, we did some analysis in our single cell data, and we want to apply a function for different clusters of cells. So instead of taking one parameter, for example, one x and one y, we can actually give a lot of x and y's to this specific content. And the last thing, which is actually important for our today's analysis, is a chain pipe, so to speak. So imagine that we again want to make this cool function. So, or no, let's take this output, and we will try to chain this. So we will not print it out. We will just use it without knowing the result, and then we will try to take it to the degree to the power of 2. So we'll make something like this. This symbol means chain, and it actually requires one of the libraries. So it will make an error right now. No, because I have this library. But in your case, it will result most likely in error, because this chain will require you to add the library here. So enter, and then you can use chains. The pipe chains means that you can actually combine quite crazy functions all together without producing any intermediate output. So if you have something as an input, you can create as long pipe chain as you like, and as an output, you will receive the something which was passing all the functions you have used. For example, we can also multiply with something, we can add something, etc. So it's just your imagination. So this very basic example is very important because we will use these pipes and functions quite a lot in the code below. So let's go to the first one and begin. So just in case if you work under, you can actually start opening the file which I provided in the GitHub. So you can open and just copy paste the very first block, which starts with fiber limit, and you see this hashtag. So hashtag means that something is commented out, meaning that if you put it here, the cursor here, and Control Enter, well, nothing happens, because for the program it's just a comment like hey, who code here. But as soon as you will delete this hashtag, you can use it. So just in case if you use Windows and your memory is not that high, I mean, your RAM is like 2 gigabytes or 5 gigabytes or 4 gigabytes or something, it's better to use this command. Unfortunately, it doesn't work with Macs. For Mac, you should have some other solution, but I guess for MacBooks, it's already fine with memory because it's at least with 8 gigabytes RAM provided right now. So if you use this, it will try to use the disk space in case if your Windows machine doesn't have enough memory. So, and I assume that all the packages were installed prior to the workshop, because we just don't have time to install them right now. So we will go further. The next step, you can actually copy paste. I will show you this file. You can just copy paste it here. Is the libraries you need for today. So you copy paste it, just put it here, and to run all the chunk, you should press not Control Enter, but Control Shift Enter, and it will run all the code chunk. And in the output, but it doesn't work on the cloud for some reason. Okay, so I will just check one thing here. There is a chance that actually sometimes it happens with the cloud. Stop.
Sasha,
Yes.
can I interrupt you?
Yes, please.
Like I see that participants initiated asking, okay, a lot of people ask something in Zoom chat, but
Okay.
because it's quite difficult to manage, I will ask you to write all your questions in our private channel. It will be easier for us to reply to you and don't have this mess. And also, if you have something really urgent to ask, you can unmute yourself or raise the hand and then unmute yourself. So it's just a small organizational issue that will simplify moderating this session.
I would actually ask you to just already tell something if you would like to. So
yeah.
Just unmute yourself. It will be much easier for me because I cannot track the chat right now. So I actually made a mistake with this chunk. So now it will work for me. So if it's highlighted somehow, it means it works, and you can Control Shift Enter, and it will begin. Yes.
Can you hear me? Okay. Yeah. Yes. I don't know what I'm doing, that I seem to not be able to use the notebook. I mean, I can use it in the sense that I write things and it doesn't do anything.
Could you share a screen for a second? Maybe we'll take a look.
Yes.
What happens.
Okay,
because it's also possible that the console, which is this panel below, just hides when you execute something, and you just need to press it again, and it will appear.
Well, the only thing that works is the part that says plot cars, that is like part of the notebook. Okay. It's like an example. That's the only thing. So I don't know if I'm writing it in the right place or not.
If you just share the screen, yes, we can take a look. Yes. Because it's important. It can be not very intuitive for new users so far, so it's better just to take a look.
Yes, sure.
Okay, this is your screen. Perfect. All right. So what you should do? So you're writing outside the chunks. So every code you actually produce should be starting with this, as I show on my screen. Okay, you don't see my screen.
Okay, so, I will stop sharing. So
yeah. So this is very common problem. Don't worry about that.
Yeah.
So any code you write with R in the markdown should be starting with this and closing with this. And if there is an unequal number of the closing or starting things, for example, I will just add an extra one. That means that your notebook is screwed, so you cannot actually write any more code. So make sure you have the equal numbers of opening and closing things. And just to create a new one, I will remind you, Control-Alt-I, and it will create the new block exactly where your cursor is located. So delete everything outside the chunks and just make it.
Control-Alt what?
I. So Control.
Okay. Okay, thank you.
Yeah, if somebody from the team could write the combination, maybe it will be useful, so we'll not forget it in a few moments. So yeah, and you can actually insert the first chunk and just copy paste all the libraries from the document I provided. And then when you did it, if you will execute Control-Shift-Enter, it will run all the chunk. So you see in my output, it already runs the first library, second, third, etc. So some people are very scared of these red lines. Don't worry about that, because actually it's not an error, it's a warning, because there's a bunch of libraries actually used, and some of the libraries share some functions or just hide some functions for themselves instead of other libraries. So it's nothing to worry about. As long as your libraries are all loaded, it's totally fine. And I just provided this small chunk also in the code. So just here, you also can make a new chunk and copy paste it. If you execute it, it will just print out like in the console here all the packages you have. So it's not like you should just recheck everything. But if you see something very similar to this output, then probably you're totally fine. Okay, dokie. So the very first step will be to understand
Again,
yes?
sorry, I'm working in a Mac.
Sure.
In the Mac is also Control-Alt-I?
So Mac users, Marina or Władysław, what's your favorite command to create a chunk?
I operate with visual interface. I will try.
I wrote it in chat. Usually for me works CMD + Alt + I.
Again, what? Oh, you put it in the chat.
Okay, so thanks for this one. So I think this command, the second one,
Command-Alt-I. Okay.
Yes, should be reproducible for Mac users. Alrighty. So before we begin here, I will just shortly introduce what we are working with. So first of all, we'll be working with liver, and this is one of the examples, but I think I don't know. All right. So this is a histological representation of liver. Actually, it's a slide scan. So it's a scanned slide of the histology slide stained with hematoxylinosin, something very, very often used in the pathology departments. So there are some vessels. So for example, here, and there's a lot of cells around vessels, and you can see these white spots. We'll magnify it a little bit more. So for example, this one, if you see on my cursor, is not they're just surrounding this big vessel. These are microvessels. So what is important? These microvessels contain small cells, endothelial cells, and after endothelial cells, immediately you can see hepatocytes, which are the most abundant cell populations of the liver. And sometimes in these vessels, if it's not a pathological slide, but in this case, it's actually a pathological slide, you can see the infiltration, so small cells with dark nuclei, which probably indicate the immune cell infiltration, in this case, with cells we cannot identify microscopically. So probably it's a neutrophil, but we cannot be sure. And actually, there's quite a lot of dark spots, which means that this is a quite severe infiltration. And since it's a viral hepatitis scan, we can assume that it's true. So what is important that I actually took a few studies and a few data sets and prepared them specifically for this workshop. And now I will show you how the data looks like originally. So we will be talking about two data sets, and we'll begin with this one. So the first one is from the healthy liver, and you can find this in the folder healthy. This is the first single-cell liver atlas used, published by Sonia McParland and colleagues in 2018. So what they did, actually, they took the resected liver pieces from the human livers from the dead donors. So they made a perfusion and digestion of this tissue. So they separated the tissue itself to the separate cell populations, and they actually just submitted the whole cell suspension to the sequencing with a single-cell RNA sequencing technique. So it briefly was described in the previous lectures. So that basically means that we had a liver, a human liver. We prepared a small cell suspension. So I don't remember how many cells were inside. And they didn't enrich them, the suspensions, for any cells specifically. So all the cells which were present in the liver probably were present in the cell suspension as well. And in the sequencing itself, we can receive very heterogeneous cell population. Another study which I used for preparing today's data sets is actually a study by Ma and colleagues from 2019 about the hepatocellular carcinoma. So here they made something quite more sophisticated with sorting. They sorted for CD45 cells. What does it mean for us? CD45 is marker for the myeloid cell population. And if you want to enrich your cell suspension for specifically immune cells, you would probably make this. and probably something else additionally, but they just separated their cells for CD45 plus and 45 minus, and those cells which were CD45 plus, then they submitted for the next single cell sequence. So what I did for this workshop, I uploaded this data sets, made partly the same analysis we'll discuss today. Yes, it's very true. As Jan wrote in the chat, it's a pan-leukocyte marker, meaning that all the leukocytes will be in the sample. So I subsampled these data sets for reducing the space they take on our repository, and also I cleaned them a little bit so it will be possible to work with these data sets without very big time spending on the technical steps. So with this understanding, we at least know that now we're working with liver. Now we have two data sets, one with hepatocellular carcinoma, another one is health. So here we can switch back to the workshop. So first of all, the very first non-package related stuff I do is I define the command here. And if you did everything as I told in the beginning, then we use the new project and not just launched some script, but you started RStudio, launched a new project, and only then opened the script or just created a new one. Your here will identify like, okay, so the output is your path to this file, and you're in the project folder, but it also can be named differently in your case, depends on how you name it. It's important because after this initiation with the here package, it will actually find all the steps, all the paths to these data sets. And you can see that we then use data directory to find the data sets, and we define the variable data dir1 as function here, which contains the folder data sets and HCC. Why is that? Because I have my project here. So this is my project folder in the project. And also I have the folder data set. And if I go there, I have two folders, hepatocellular carcinoma or HCC and healthy. So this is the structure which you should have in your project as well right now. If it's not like that, then probably something was wrong and you should return back and restore the proper course. So nevertheless, what we will do, we'll execute this chunk right now, and I will explain what it does. So Ctrl-Shift-Enter on the chunk, which you also should copy-paste from the. So somebody asked on the workshop channel how to find this code. Actually, I wrote a few times, so it's provided on the GitHub repo, and I copy once again the link. Please just copy-paste the chunks from there, and we will proceed with this code to be copy-paste. So what it does, it actually takes the data files. So we'll go to the data sets and I will show you one. Each data set contains 3 files, which we discussed, I think, yesterday in the Marinos lecture or the day before. I don't remember it now. So after we sequence something, right, and from the Illumina sequencer, we receive the BCL file. If we use the 10x single cell sequencing, we use specific software which names Cell Ranger. And from this BCL file, with a few comments, we produce these three files: barcodes, genes, and matrix. So now is an important step to make the specific object which can be used then in our next analysis. So I will go just back a little bit to the code, and I assign sample1 data and the function red10x. So red10x is a specific function which goes to this folder where the sample is located because we pointed it there, and seeks for these 3 files. And if these 3 files are not here, you will receive an error. If it worked, you will receive, I think, nothing. Yes, there is no output. And then when we actually uploaded these 3 files, I use the next function to create Seurat object. So Seurat is a package which is very commonly used for the 10x related single cell RNA sequencing analysis. It creates a specific and very nice object containing all the necessary data about the cells, about the genes, about the barcodes, and we will just take a look in a few steps. How does it look? So, and I do this separately for the disease sample, HCC, and I do this separately for the healthy sample. So basically the same pieces of the code, just for different samples. So now we have two Seurat objects, and now I think it's a good point, and we actually can see how does it look. So if we now write, after we executed this code, if we write the first object's name, sample, we can go inside this object and take a look. Actually, we will do it a little bit differently. So in the upper right corner, usually you have this environment window open already. In my case, I have a lot of stuff already open because I've opened the cloud and everything is already loaded in my session, but you will only see the HCC sample, healthy sample, and a few more variables in this global environment. So let's find HCC sample and press on it. After you press on it, the separate window will open near the script. And here you can see the structure of this object. So let's see. Yes, so it's a new object. We created a new object. What is important here, and it's quite sophisticated object, but what is important to know is metadata. So it's something you should be aware when you work with some Seurat object. What is actually in the metadata? So first of all, there is this original identifier, which you just put in the code, just here. So project equals HCC or project equals healthy. And there's many ways how to identify what is inside the object, but I just use the original identifier. So it will always be marked with HCC, and every cell from this object will be marked with that. So I will just know that it arises from hepatocellular carcinoma sample. Then the M count and M feature. So this I will explain a little bit later. And that's actually all we have at this step with the metadata. Later on, we will add something else on the metadata, and actually metadata is quite a heterogeneous, I would say, section of this object, because different people put different stuff here. For example, you can, identify your cells not with original identifier, but add another column like cell names or cluster names or whatever. Next, what is important is assays. So right now we have the RNA assay, and it's and the count matrix, which we'll also observe in a few moments. So when we'll make some few further analysis, when we normalize the data, we'll always also see the additional assays here based on the normalization method we prefer, because the original count data will be then used and modified by the normalization functions. And also you can see here neighbors, reductions, and images graphs. So when we do, for example, the dimensionality reduction discussed in the previous lectures, it also will be saved in this object. So we don't have to create different objects. You actually contain everything inside one Seurat object. And we will step a little bit further because I actually would like to show the integration of Seurat objects here. So that's why at some point we will integrate. I just think now, what else can I show you the Seurat object before we really begin? I think it's pretty much everything. Okay, so first of all, we'll try to make some quality control. As you remember, a lot of things should be normalized to something, because I think either Vladislav or Marina already discussed that. Yes, so it was yesterday during the Marina's lecture. We discussed that mitochondrial content would be a good parameter to normalize for, or to filter out the outliers, because it indicates quite a lot regarding the cells. So, for example, if the cell is suffering, agonizing, and almost dying, or there's not that much of mRNA left on the cell, so, for example, in this case, we will see a lot of mitochondrial content, like 70%, 80%, and most likely these cells are not what you would like to receive in your data set, because the remaining RNA could be related to the reads from the pathways related to the cell death, for example, or some heavily pathological changes related to the sequencing procedure itself. However, it's also a little bit tricky. So if you will find the manuals regarding the filtering for the mitochondrial content, most likely you will see the PBMC's manuals, which show you that actually you should cut off every cell which is which is higher than 50% mitochondrial content, which is also true, but not for every tissues. So for neuronal tissue, and probably I'm not for sure no, but we discussed it yesterday, and definitely for liver, you don't want to apply the same filter because it's too low. Because there are normal hepatocytes, for example, or normal other liver cell types which may contain up to 30-35%. However, we should first calculate the mitochondrial content, and actually it uses one of the inbuilt Seurat function, percentage feature set. And we just set up the pattern because all the mitochondrial genes start with empty in the count matrix. So let's do it for healthy and diseased. So we'll do it like this. Enter. And as an output, probably below the code chunk, in my case, it's in the lower right zone with plots, you will see two beautiful quotes, one for healthy because it was first, and one for disease. So as I told, it's not, so you can already see the mitochondrial percent, which was added to the metadata by the code we just executed. You can actually see that these data sets are quite different. So for example, the healthy, I think it's healthy, yes, the healthy data set contains like cells from almost 0 to up to 30-ish% mitochondrial content. If we'll take a look at hepatocellular carcinoma, you will see the cells that do not contain that much of mitochondrial content inside. So usually it's less than 10%. So now the question to you, Zoom participants, why do you think it's that? Because, you know, it's all two samples from the liver, right? It should be maybe more similar, or maybe not. Any thoughts?
But one is diseased, right?
Yeah, definitely. So this one is diseased. It's from the diseased liver. And this one is from healthy liver.
Could it be because they are diseased? I mean,
Oh.
they have a disease, and that's why they have a higher content of mitochondrial genes.
That's actually a very nice suggestion, but and probably it actually may impact somehow some of the data sets. But here the trick is in the data preparation I showed to you, because the liver from the hepatocellular carcinoma, not actually all the cells were used for the sequencing, but only the immune cells. And immune cells a priori contain just less mitochondrial content compared with hepatocytes. So that's why we see a lot of mitochondrial content here in healthy liver, because there are a lot of non-immune cells as well left, and very, very limited number of the mitochondrial content here, because actually there are just immune cells. So that's why you should just keep in mind, if you work with public data sets, it's very likely they were prepared with different protocols, which may include or not include some cell types. And that's why you should consider that when analyzing stuff. However, the last part of this chunk is filtering out, and you see what we do here for both examples. We use a function means subset, so it just selects a part of this of the data set based on the certain functions. So in the first case, we subset what was and most important stuff regarding the functions in R. If you write some functions, yes, like this. So it can highlight itself and show you what it does. And sometimes it shows you what actually should pass to this function. For example, subset, it's not really helpful in this case, but X is the data set you should pass, and this 3 dots is all the rest of the arguments you use. In the case if you are struggling with something, Jan writes the directory provider doesn't exist. Please make sure that you created the R Studio project, not just a script, and in the same folder where the project is located, you can find the datasets folder. So you see, here's my project, and here's my dataset folder, and everything is going on inside the project folder. And this dataset folder should contain only these 2 folders. So it's not excluded that you just unzip it incorrectly, and inside the dataset folder, there's another dataset folder. Yep. So, and in case if you're really struggling with some functions, you don't understand what to pass to the specific function, like I write my awesome data set and what else. So you can Google this function and the very first results you will find related to it, and you will find the list of the arguments. So for example, let's actually try it. Seurat subset. Our documentation, subset data. Okay, so it's not exactly the same function, but I think if we Google a little bit more, we'll find it. It shows you, okay, so for this function, let's say subset data, you should pass the object, so it's your data set, and you should pass some additional arguments. And if you're still not sure what does it mean, so cells, what is that? You can go below and find, okay, this is a vector of cell names used as a subset. For example, if there are a vector of cells already named as T cells, you can just write it there and quote it and subset only for these cells. It's a little bit different function, but still it's how you can find some information. Okay, so let's turn out, filtering out. We subset from the object named healthy sample, which is our Seurat object containing all the healthy cells sequenced. So we subset based on the feature and feature RNA. So I promised to explain you what does it mean. This is a very important thing for the quality control. So This feature RNA itself is the number of genes which were detected in every cells. And why do we sort for cells containing more than 200 genes detected? Because we assume that the cells which contain less than 200, either weren't sequenced properly, or maybe were dying, or maybe this gem bubble which is used for sequencing, maybe it contains just remainings of the cell, like something, after the cell death. However, it's totally arbitrary, and it may be different for your specific analysis. So you can either use standard features, standard values provided by the manual, or just play around, and find the best solution for your data set.
That's what I was going to ask you. How do you decide that? Does it have to do with the number of cells that you are?
No, actually it doesn't. So what I do usually the values provided in the manuals are totally meaningful, because we select for everything with genes sequence more than 200 and below 2,500, because if we will think about values up, higher than 2,500, probably it's a cell doublet. So if you see some cells with like, so to speak, cells, right, with, I don't know, 7,000 gene sequence, probably it's just a doublet.
May I say something to this, if you
allow?
Can you please scroll up to the plots, to the violin plots of the N features?
Sure.
So maybe because for example, for me, it was also a very crucial question, as you put it, Carolina, to how to decide what is the quality control on yourself. And as Alexander says, like this 200 and 2,500 are good values, but it hardly depends on the data set that you are working with. For example, if you are working with the T cells, or if you're working with the other lymphocyte cells, it means that you will expect the number of genes around mean like 1,000, 1,400, etc. And if you're working with cells that are myeloid cell populations, like very big cells also that contain a lot of the RNA, or for example, with plasma cells that are producing antibodies and have also a lot of the RNA, you would expect even more genes in each of the cell. It can reach even 4,000 and 5,000, and in that case, it would be okay. So, when you are speaking about the lymphocytes, in that case, 4,000 may be doublet, but when you're speaking about the plasma cell, 4,000 may be okay. And that's why the best way to set this cutoff is just to look at your N feature RNA. And when you see the violin there, you should point the upper point as the, let's say it's 2 or 3 times more than the mean of the main violin. So in that case, when you have the mean around 500, maybe 700, so the upper point would be 2,200, 2,500. So that means that you should just cut off the upper outliners on the violin plot, and it depends strongly on the data that you are analyzing. Your violin may seem completely in the other way when you're working with the other project, but just keep in mind you have the mean and then maybe 2-3 times more, and then cut off on the top. So that.
Yep. And also another trick, if you see any papers related to your topic, just take a look what they do. Sometimes it's not really helpful because some people sort it totally differently. So for example, as I mentioned yesterday, I think the very first papers, Seurat and Lieber, were sorting, so they filtering out all the mitochondrial content higher than 15%, which according to today's understanding is not, that's the best solution. However, it can approximately give you initial insights on how the people sort the issue. However, if you're the pathfinder and you're the first to discuss something, of course, it's always helpful to actually take a look at the plot and see where the majority of the cells are located. Still not the perfect, I would say, thing to consider initially or ultimately, but it can also give you some insights. Yeah. Actually, what is important, you can actually see that there is not that much of cells higher than 2,000 reads, and unfortunately we don't see the scale, but I think that below 200 it's quite some cells, but probably but in this case, in this healthy sample, at least you can see them, most of them are below 2,000, and in the diseased sample you can see that they're somehow spread, but It's also, again, it's possible just to calculate it more precisely. Okay. So we make the filtering. And
Alexander, can I ask you a question?
Yes, please.
Yeah. So I was looking at Rahul Sathija's Vignet for Seurat, and they consider 5% to be the mitochondrial content cutoff. Do you can you suggest what should be the cutoff when we are filtering or subsetting the data?
So it's again always very tissue specific. Why do we know that, for example, for liver 30% is fine? Because at some point, I don't remember when, I think it was 2018, some paper just showed up describing the donation of hepatocytes in different parts of the liver. And it was specifically pointed out that in some parts of the liver, very normal, healthy, not dying, not apoptosing cells contain approximately 30-ish% of mitochondria. That's how people started to sort up to 30. They elevated the threshold up to 30% from 12 to 20 or 15. But for your tissue, it can be totally different. Here, I just propose to use the arbitrary 30% and see what happens. Don't forget that even if you are not happy with your result, you can always go back and sort differently. So if you don't see any good outcome.
And a very, very good example in that case would just to look at the violin plots once more that we had. And on the right, you would see the %MT. So as you see, you should take a cutoff that will have the major population of the cells that you would have in your sample, but remove the outliners that are on top, and that would be probably dead cells. But as Alexander already pre-qualified the data set, so you said that you already cleaned it up. It means that we don't need to clean it up too much further.
The question is regarding why do you see the plots not in the separate window? There is this specific view button, right? And you should just point show previews inline. And it will just stop showing you the previews here under the code and start showing it in the lower right corner. But again, it's just up to you. Some people just enjoy seeing immediately below the code. So yeah. So now what we did, we made the initial quality control there. We think we're happy with these numbers, and then we will do the data integration. So what does it mean? We have two separate data sets, and what we could do, actually, we could just, go independently for both of them, and, just to identify all the cells, then to mark the cluster, etc. But I see a good reason to integrate the data sets, so we can just do it once. And we identify all the cell cluster, which are, split between these two data sets in just one integrated data set. So what we will do now, we'll create the list containing these two variables, healthy sample and HCC sample, which we received in the last code. So we will make this lapply, which just applies this function to the bottom of the elements. And we'll do next. So for me, it's just, 1 second. So you see differently because I accidentally, minimized the code chunks. So you see it like here. Yeah, let's actually do to the both elements of the list, we apply this normalized data function and then find variable features function. So fortunately, nowadays, we don't have to see and play with very much with the arguments which are provided inside this function. So more or less for the initial analysis, you can select this example. However, what is interesting here, I used the second option. So what it does, before actually executing the integration, it does the normalization of the data. In this case, with option number 1, or just data integration, it used the log normalization. So it's a not very resource-dependent thingy, which scales data by the library size. So as you can imagine, all the cells are sequenced separately, and that's why they had the separate, the different sequencing depth. Meaning one cells could be, could have 100%. If, for example, let's normalize, one cell contains 100% of reads, another cell contains 300% of reads, the third cell contains 50% of reads. And it's just not very, very good practice to compare the cells without prior normalization. So that's why we actually do this log normalization. So we scale the data across all the reads. So the future count of every cells in the data set is divided by the total counts for the specific cells. then it multiplied by specific factor. So I think for the log normalization here it's 10,000. And after that, it's then converted to the natural logarithm. So just transform it as natural logarithm. That's why it's called log normalization. However, what I did, I also put the second option here because for my personal analysis, I don't use log normalization that much because there are better options for data normalization in my data sets with liver. However, it's very resource dependent, and even 8 gigabytes of RAM should be not enough, or usually it's not enough for such normalization. The name is SC transform, and there's a paper in I think 2019 published about that. So it's a little bit more, how to say, more sophisticated method. So it's using something named regulated negative binomial regression. So it's more resource heavy. So it uses similarly abandoned genes between the cells. So they use this as covariates for the model in the regression. Usually it works good with small data sets with at least 16 gigabytes of RAM. That's why it's actually commented here, and we will not try it. Otherwise, it will take all the eternity. So, but it's actually very simple. Oh, sorry, very similar to that. So we will use the log normalization, and if you enter the code, Control Enter or Shift Enter on that in the console, you'll see the similar output. So it performs log normalization, then calculated gene variances, and well, after this, for the integrated data set, it's normalized. So it also finds the integration anchors between the two data sets and just integrates it based on the log normalized values. And now the count matrix is significantly modified, so we don't have any absolute counts anymore, but we have the log normalized counts, which means that we have now two assays. So we have created this data sets integrated object, and if we'll go to the metadata, data sets, integrated object, and let's go to the metadata. Here you will see, Here you will see, first of all, we have a new column which we created before, percent mitochondrial content. But importantly, we have the original identifier. And here you see healthy, but if you would open it widely enough, deeply enough, we would see hepatocellular carcinoma at some point. So based on this original identifier, we can now separate which cells came from which data set. So that's what I'm going to show you. And we will not use this data integration option too, unless you work on the very, very powerful PC right now with a lot of RAM, and you just want to make it in the parallel. Then just please tell us, and we can compare. However, another topic, or so to speak, the next step we should usually do in the standard workflow is to reduce the dimensionality, meaning that we have this still even normalized. We had the big matrix with genes and the cell names, right? So I think we can actually try to take a look at that. And if
Alexander,
yes,
me again.
Yes,
so
please.
I used the datasets integrated at metadata, and I had like object dataset integrated, not found. What did I do wrong?
So it means that probably this chunk of the code above wasn't executed for you, because if it's executed properly, it just creates a new dataset integrated object with this integrated data. Did you copy paste this chunk from the file?
Yes, I did. I am following exactly the. So let me see if I just.
Right, but did you see this output in the console I'm showing now? So this is red lines.
Yes, I have those.
Yep.
Yes, the red lines, I have them, and what did I do?
Then maybe just select again, this later parts, and execute them again.
Okay, I will do it. Okay.
Maybe you skipped one of the lines, so it happens in this case.
Oh, okay.
So in this case, what else is important? Okay, so now if we'll take a look again on the Seurat object. So right now it is integrated, and we'll go to the assays. And now we have two assays. First, RNA, which is non-normalized counts, like the absolute count numbers. The second one is integrated. So yeah, and if we will take a look right now, so assays, and let's go to the counts. if this cloud will not explode, no, actually it should be something. So I just wanted to show you the matrix itself, which is quite big, and I'm not sure whether I will be able to show you. Yeah, probably it's not the best idea to show you. Okay, nevertheless, so you just have to believe me. It contains the matrix with genes and cells, and in the crossing of the genes and cells there are values which are either 0 or not 0. So, however, we cannot really plot this data because it's like 20,000 dimensions or per cell, which is quite a huge number, and we have probably a few thousands of cells here. So what we'll do, we will scale the data and make the dimensionality reduction based on the principal component analysis, and on the top we will add the UMAP. So UMAP is actually quite an interesting thing, which we even plot right now. So I propose we just include it. All this. So the second part of this is hard commented just because we didn't use this SC transform as I mentioned. We use the log normalize. That's why for that we use this part of the code. Because if we use SC transform, we don't actually have to scale the data because that's how the SC transform works. So it's still very similar with PCA UMAP, and it just doesn't use scale. Again, if you're not sure, you can either search for the function or go to the Seurat manual and find this. And in the output, after the find cluster, the last one, you will see the small notification that perhaps 10 seconds, meaning we're done, and the number of the communities. So this is the number of our cluster which were detected. And right now, yes, so the next chunk of the code is actually plotting, and here we will start producing the fancy plots. So I propose to execute it. What we do, we create two plots, P1 and P2, with the function dimplot, which is an inbuilt function in Seurat. We plot the data set, we select the reduction UMAP because we applied UMAP, and we, in the first case, group by this original ident. If you remember the metadata, the original ident is something which is either healthy or hepatocellular carcinoma. And in the second case, we do something differently. So if I may, I will just show it here, and probably you should also receive these images. So this is called UMAP, and it's the dimensionality of our data set is reduced to the 2D, so perfectly visible for humans. What we see here on the left, it's a plot number 1. So we have split our UMAP to the original ident. Sorry, grouped by the original ident, and we see one is hepatocellular carcinoma, which is red, and another one is healthy. A good thing that cells are actually plotted very close to each other. So you can see that this in the middle, for example, 2 clusters or maybe more than 2 clusters are plotted very close to each other. And in general, the clusters are overlapping very well. However, sometimes the clusters are not exactly overlapping. In this case, you should think whether actually your data set contain a lot of similar cells or not. But however, here you can see that, for example, in red, hepatocellular carcinoma color in our case, we have a lot more of these cells as compared to the healthy control, but we still don't know what the cells are inside. And another one just splits according to the clusters. So we have detected 18 communities, as it was writing to us before, and we have here from 0 to 17. So in total, it's still 18, but just starts from 0. And here we can see how many communities we have, where are they located. For example, these ones are located quite close to each other, but still 3 different clusters according to their anchors or expression features. Here again, we can see quite a lot of different cells. However, we can also do something different. So for example, we can change group by to the split by. See what happens, because it's also one of the standard and probably we actually don't need a second plot. So, yeah. And here you can see in the previous case with the group by, we've seen them overlapped overlapped one on top of the another. Here we can see them separately. So the left part is for hepatocellular carcinoma, the right one is for health. It may be not that helpful. I personally might like overlapping more, but if you want just to plot something for your paper, or just to observe a separate data set, please feel free to use this function argument for the function as well. And in general, this gene plots or this logic of showing the plots like P1 plus P2 will show the plots all together close to each other is taken from the ggplot2 library for R, which is very famous for plotting, meaning that, you can technically take all the data sets from the Seurat and, make any plot you can imagine with ggplot2. However, it may take a lot of time if it's something crazy enough or something nobody ever done before. So that's how we use these plots. So are there any people struggling up to this step yet? Do you have maybe any questions or something doesn't work for you very well? Just identify yourself, so we will need that. We will know that. Okay, all good. It's actually a very good sign. Super, thanks. So next is actually will be a little bit, I think, interactive. So, okay, yes. So this step will be interactive. So we have 18 clusters, but we have 0 ideas for that. So I will ask a few persons, if it's possible, to help me a little bit. So I will ask Marina, Andrea, and Tatiana. So please open Google, and I will ask 3 of you to Google for human cell markers for specific cells. I will ask you. So please, Marina, Google Kupffer cells. I will write you how does it.
Okay, I got it. Yeah.
Kupffer cells. Please, Andrea, Google T cells, and Tatiana, please Google B cells. So just tell me a few markers you will find. So if you Google that, like B cells, human markers, you will see some papers in the very first results, which say, okay, so this gene was detected in the B cells. Just name a few of those to me, and we'll try to plot it and see where we probably have the cells. This is the number 1 option how we can identify these cell clusters. The number 2 option will be a little bit more interactive and fun, so it will follow after. Yeah, and while we are doing so I think we have 2 minutes for that. There's a question: Does it also plot t-SNE? So t-SNE, like small t and big S and E, is another approach for dimensionality reduction. However, today UMAP is the most widely used for such kind of data sets. Unfortunately, I'm not that smart in regard to the dimensionality reductions. I know that there are crazy mathematicians working on that, and can tell you very deep math related to it. But I just know that UMAP is somewhat more trendy right now in such kind of data sets. So there's a lot of ways how you can reduce the dimensionality, and UMAP is just the standard way, the first step you can use. And I honestly think in the most of the single cell RNA-seq data, it will just perform very well. Oh, do we have something?
Even from the marker genes?
Yeah, just name it please a few. Oh, okay, thanks. So Tatiana sent me a few markers. I will just try to. Okay, I now wondering. Thanks. I also have a few from Andrea. Now I'm not sure whether the genes actually. The gene names are actually the same, but we'll just try it. And if it doesn't work, it will tell us, okay, so CD27 minus 1. And we'll see if the gene names are different, and it will just show its error. If not, then because not all of the CD genes are actually also named CD. So from Andrea, we have.
Sorry,
yes?
PDL1 plus.
Yes, I know, but the gene name is definitely without the plus. So, okay, so IRF4 is definitely a gene name, and remember that. IL9. So usually how I do this, I go to the public database, but just for fun, we go to this way. CCR6, reticulin beta 1, and the last from Marina, C1QA.
I literally do not know. It's just like a meta-analysis.
And QEC4G. QEC4G is definitely it, and marker is definitely it as well.
Okay.
These genes I remember. So, okay, now we have the markers of interest because, we were super smart. We made a meta-analysis of literature and just, have a notebook with the markers we need. And first thing we will do is to change the default assay from the integrated one to the RNA one. Because when we integrated the data set and normalized the data, we lost a little bit of biological variance. And we still want to define the cells from the biological variance. The first point. So we will make default assay RNA, and this is just the name of the assay. And the second name is integrated, which we can take a look in the Seurat object. And then we can define the list of the features. And the crazy plotting begins. So this is just array containing the gene names. It can be whatever you want. So first of all, we will use the feature plots. Feature plot is a very standard way in Seurat how you can check where the genes of interest are expressed. So we will use a dataset integrated features equals feature list. And this is some more sophisticated things you can read about. And label is true. So we execute this code. If you think a little bit, depending on how powerful is your laptop. Okay, so it actually prints a few errors. So they didn't detect CD21, PDL1, and CD43. And PDL1, I think, just because I'm very stupid and it's without this number symbol. So it will definitely tell you that from this list something was not detected. However, a lot of stuff actually was detected, and we will take a look right now. So let's go first. Marina, you had Kupffer cells, I guess. Let's start from the end. So Marco, and that's just I know because I work with Kupffer cells, is a symbol of non-activated, is a marker of non-activated expression of non-activated Kupffer cells, which are liver macrophages, and they're very much expressed in the cluster 15 and 6, which is quite interesting, and a little bit expressed in different clusters as well, but that's not that interesting for us at this point. The next marker is CLEC4G, also identified by Marina, and this cluster, reverse is not expressing in this 15 and 6, but might expressed in this 9, 1, and 17. So probably some macrophages are present here as well. And the third one is PPARD, which is not expressed that much actually over the data set. But one of the markers, C1QA, which is also a specific marker for Kupffer cells, is again expressed in the clusters 15 and 16. And 6. From that, we can actually already make a hypothesis that probably these clusters are actually Kupffer cells, which we looked at much. But still, it's not sure what happened there with cluster number 9, why these clusters are also positive for this specific marker. Probably the answer is that this marker is not that specific, or probably it's expressed in other macrophages as well. So we will see it further. Let's say from, I think, interleukin 9. So it's from the Andrea data set. Did you study B cells or which cells? Could you remind me? Or T cells? What did they tell you to find? Okay, if Andrea is not here, because we can hear her.
It's T cells.
Yeah.
T cells. Okay, so we can see it's super non-specific for our data set because actually all of the cells expressing it. However, if we will go further and find CCR6, I think yes, CCR6, which, as according to the paper Andrea found, is specifically expressed in the DC17 cells. We can actually find a small cluster 3 with quite some expression of this marker. So probably these are TC17. However, it's not for sure now. And what I definitely like more is this KLRB1, which actually expresses here. So I would assume somewhere between these clusters you can find some T cells. And from the sample which Tatiana received from these cells, unfortunately I don't see any strong expression of these markers. However, cluster number 13 and clusters number 5 and 2 may contain some B cells. However, it's not for sure. And again, it's the first marker we actually do, so probably it's not the perfect markers, but still, for Kupffer cells, I'm already quite confident. And technically, if I already know what we're looking at, we can just name, okay, then cluster 15 is Kupffer cells. However, now we do something more interesting. But before, we'll just make more crazy plots because actually they look nice and you can see them all over the papers. They're quite standard. We use we will reduce the features list and make the rich plot. So that's how you will just see how it looks.
I also would like to add that there is other way to do these observations. I mean, you can start by plotting these genes, but what I usually do, I check this table where these marker genes for particular clusters are already presented, and by
it's step number 2. We'll go for it.
Yeah. Okay. Yeah. Good.
No spoilers. So the rich plot is also a nice and standard plot you can use from Seurat. Not for me, it's not that much informative, but still you can find it. For example, PPA-RD marker is not exactly the most popular marker in our data set. However, interleukin-9 is super widely expressed across all of the cell clusters. Yeah. Another nice plot is violent plots, but again, it's nothing interesting. Just usual violent plots. So So these plots depend on which way of selecting or highlighting specific cell markers do you prefer. These are just usual violent plots divided, so expression divided by different cells. Of course, you can also separate them if you would like to, according to the healthy versus hepatocellular carcinoma, or use this dot plots. So it's like your personal preference, and the style of the figures you prepare for the publication also, of course, impact your selection. All right, yeah. So now we will go to the exactly mechanism which Marina discussed and which I actually used in the real life the most. So, but we will not execute it right here. Instead, we'll go to the GitHub and open the first link, annotation markers. So why we do this? Because it actually takes a little bit more time. So first, you're free to go to the GitHub and find the markers, and I will just show it also in the Zoom chat. So what I do here, I don't actually remember. So I think at some workshop or some fellow bioinformatician advised me that it's not the only way how to do this. However, it's how I usually do this. So I connect to the annotation hub database, and you can then, if you would like to to make the autopsy of this code and just to investigate every block, sub-block. But what I do, I actually go to this public data set, I request the information, the descriptions of the genes I found, and then I perform the usual find converse markers function for every cluster. And then I just set them to the table. So this is it. And in result, I receive such data table with p-values, adjusted p-values, principal components, which is not that important for us, clusters, and the genes, and genes is and the descriptions of these genes. So my cool cells is actually the cluster number 0, but for the preparation of this workshop, I just named them my cool cells. So it's cluster number 0. Then we will do something which I show you right now. How I do this usually, there are numerous public data sets, and one of my favorite ones is the protein atlas. So we'll open it right now. And in such public data sets, as for example, the human protein atlas, you can find where the specific marker is expressed over the data set, over the cell types they have. So for example, we'll go to this data table and find, okay, micro cells express very much of S100A8, right? So we'll just copy paste it, go to the protein atlas and take a look. So here you can see the expression in the different types of cells. So for example, we're the most interested about the cell type because it's the tissue single cell data sets that they have, but also since we have a lot of immune cells infiltrated in the liver in hepatocellular carcinoma, for example, sometimes we should also take a look in the blood data sets, just because these cells were originated from blood. So we will go to the data set based on the cell type, and we will go down to the liver, and we can sort by the expression. And you can find that, actually, Kupffer cells, one of the clusters they have, the database expresses quite a lot of this marker. There is some expression on the other cluster cell Kupffer, but very minimal one. And there's a lot of cells not expressing them at all. And if I would check a few more markers, just to be sure, for example, this, I will do basically very much the same. We just please for the lysosomes, I will go to the cell type, and again, I'm mostly interested in liver, so I go to the liver. Again, Kupffer cells, and exactly the same cluster we had before, and a little bit in other cell type. So after I check this, a few more, I just already know from the previous analysis that these are Kupffer cells. However, it's most likely we have several Kupffer cells clusters. So I will copy the first few markers, for example, and I will use the function rename idents. So in my case, it's cluster 0, and I will write Kupffer cells S100A8 plus, this plus, just because different Kupffer cells cluster may have different markers. Yes, Camila, this workshop is recorded on YouTube, so if you would like to check it later, of course you will find it. So now, what we'll do next in your anonymous window? I think here, for example, I will try to open it myself as you open it right now, so without any authorization before. So I will ask you to go to the clusters table, then tap data and select. Yes, so on Google it works like that: data, filter views, and create new temporary filter view. So it will create a filter around the cluster. So the good thing would be if we would distribute the clusters now, and we'll try to mark as many clusters as we can. So this filter appeared, and you can, for example, clear all and select only cluster 1. And again, here you see cluster 1 and the genes checked as the most because the adjusted p-value is the least in this cases. So what I ask you to split the clusters, and I will just name a few names right now, and it would be cool if we will do the same, and then it will just tell me which cells did you find. It's on you, so I will not recheck it, but I remember a few clusters, so I will be able to immediately tell you in a few cases if it was right or wrong. So I would like if Carolina would like to make the cluster number 1, Dipanjan cluster number 2, Andrea cluster number 3, Tatiana cluster number 4, Irina cluster number 5, Jonathan cluster number 6, please, Taras 7, Temur Khan 8, Igor 9, Julia 10, Behnam 11, Preeti 12, Pierre 13, and if there's any, yes, of course. So Veronica 14, Akembo 15, Meha 16, Paula 17, and I think we have one more cluster which goes to Roxane. Then I will ask again Denis to help with cluster number 1, Muhebat with cluster number 2, and IMS. Unfortunately, I don't see the whole name. It's cluster number 3. So I think it will take us, let's say, 5 minutes. So if you're watching us via YouTube, you can go and make some tea. Otherwise, from you, I will ask you to provide me in 5 minutes the name of the cells you think are the cluster, and maybe a little description why do you think is so. Or so maybe you can also Google what this gene means and what it does, because in some cases we will find different clusters with T cells, for example, the activated T cells, the TC17 T cells, some memory cells, etc. And it's important to distinguish between them. So, and if you struggle with something, please also immediately unmute yourself. So we can. And in the meantime, I will just check the messages on the Slack. Maybe we have questions. Okay, so there was one of the questions regarding the PCA. It's not, so why do PCA before EMAP? Does it override itself or not? No. And actually the PCA is an initial reduction, which is important to pass to the UMAP. So UMAP will use it as an input. So no, it's not overriding itself, it's overwriting itself. It's just different steps of this analysis. Okay, so one question answered. Oh, okay. Thanks, Andrea. We actually have already cluster 3, and I will ask you to actually tell us what do you think according to the database, the name of the cells, so we can just write the human readable.
So it's a T cell receptor alpha constant and interleukin 32, CD3D molecule, osteonectin,
Okay,
CD2 molecule.
According to the expression in liver on blood, what do you think the cells are? I mean, so it's definitely T cells, right? Because I will actually do it with you right now, because sometimes it's not that easy to find. So we will just use the markers you mentioned, for example, CD3D, right? And we will go to the liver and to the blood. So if we will go to the liver expression, here below, we will find that it's actually expressed in some T cells. However, as we know, that T cells are not actually originating from liver. That's why we maybe also should check the blood sample. And if we go to the blood data set, where is it? So there are different data sets. We can find that actually it's very much expressed in the Tregs and NGDT cells, which is already more interesting. However, it's not that much specific because as we can see, it's pretty much expressed in many cells, including the naive cells, other type of naive CD4 cells. So in this case, it's very, the real life example. The next step would be to go to the publications on the PubMed and actually to search for every of these markers. However, for the simplicity, I would say that we'll just mark them with PCR, so these cells. You're looking CD3 plus, CD3D plus. It will already help to distinguish the cells today. However, in the real world, you would like to define the cells a little bit more specifically. So we have a question: how can we choose the right cell cluster? If we could ask something more specifically, that would be also very helpful. So Muhibat writes that cluster number 2 is probably the T cells as well. Muhibat, do you have any opinion about these T cells? Are they any specific T cells, or maybe have specific markers? Or you Google. Feel free to unmute yourself if you can, so it will be easier for us to hear. Or if you don't know, we can actually check. Okay, CCL5, again, CD3D, and granzyme H. So let's check the CCL5 in the liver. Okay, apparently liver doesn't show us a lot because they were marked always just T cells in the liver data sets. Let's check the same in the blood. So in the blood, it's either CD8 or gamma delta T cell. So there are definitely still T cells, and this one. So again, not for sure because they are very similar. And of course, we can just again mark them with granzyme CCL5, granzyme D. However, just just be sure. Yes, I don't see anything more specific here. So in this example, we can also write the markers, so we identify them later. So Taras writes that cluster 7 are probably T-Rex. Let's just put it like that. So as Mohabat mentioned, in the liver, these cells are written as C10, C15, C1, which means that in the specific data set they use for the liver tissue, they just market them as T cell cluster 10, cluster 15, and cluster 1. And people often do this, for example, if the cells are newly described and there's not that much of information regarding this market combination. Yes, so definitely. Jan writes about help. Yes, for sure. If you tell us what is your struggling with yourselves, we will definitely try to.
Yeah. Okay. I got cluster 6, but how do I start identifying?
So you filter for, as I showed. You filter for cluster 6 in the data table, which created, and then you can see that the most confident expressed genes compared with other clusters are CD5, C1QB, CTSV, etc. So you just copy the gene name, for example, CTQB, because I think CD5L will be not very specific. You go to the protein atlas and you insert the gene name to see if it's expressed in any cell from the previous data sets. It's a complement cells, which already tells us that probably it's somehow related to the maybe some specific macrophages. So if we go to the tissue and go to the liver, we already see quite a nice picture. So this marker is super expressed in Kupffer cells and not expressed at all almost in all the other cell types. So I can already tell you that it's Kupffer cell marker. And it's actually very, very nice because I know that this specific cluster, according to my previous evaluation, was up to the Kupffer cells. So we just start Kupffer 6, cluster 6 with Kupffer cells. Is it more clear somehow for you now?
Yes, it is. But let's say I'm analyzing an independent data set. Yep. Where do we get this annotation table from? The one that you gave us.
So this is a chunk of the code, the annotation number 2, I named it, and you can just execute it with your own data set. Why they didn't do this? Just because it's resource heavy, means that it will take approximately 30 minutes for us to generate this table. Otherwise, if you have time, you can just do it, after the workshop again with exactly the same code, and it will save it as a data file and conserved markers.csv, and you can open it. So it will be exactly the same file, but with different clusters if you use it in different data set.
Sure. Got it. Thank you.
Yeah, so we have a lot of inputs already. We will just mark a few more clusters and we'll go further. So cluster 11 are probably neutrophils. Works for me. Cluster 4. Okay, so this cluster is interesting. What do you think, Tatiana? Which cluster is that according to the specific cell name? Yeah, so it can be in the tissue, but which do you think is the cell name? So which specific cells are those? Well, the very good suggestion you can already have, and it's the name of the first marker, J chain. So usually these gene names are related to the either heavy or light chains of the B cells, because they produce immunoglobulins which have light and heavy chains, and probably this one is related to one of those. But just take a look. So that's another interesting example with the cluster number 4. If you will see the cell type, it already shows the most expressed one in erythroid cells, which is definitely probably not the case in this regard. But if you will check the blood expression, it most likely show us that it's expressed in the B cells. So memory B cell, plasmacytoid, or naive B cells. Why is that? because again, the cells are not of the tissue origin, and probably the data set they used for the liver tissue, it was from the healthy liver, so it wasn't containing a lot of memory B cells, naive B cells, etc., which appear in the tissue usually when there is an infiltration and inflammation process. So these are definitely B cells. We just mark it with that. Cells without further, and we will just mark those guys you already sent to us. And okay, cluster 70 mosquito cells. That's a good observation. That's very, very likely true. I remember something about this. These are liver-specific cells, but not the immune cells. Cluster 8 is Kaplan cells, other cell type. And we will finish with the last one you just seen. And this is again another mark. Okay. So now I think we will stop it right now. So you can continue after the workshop and just to finish the annotation. But to save some time, we will just stop here. And I will make the function which is called rename idents. So I will rename all these idents from 0, 1, 2, 3, 4, 5, etc. to the names we give it here. However, what is important for you to know, it's not the only way how we can rename the cells. It's also possible to leave this cell idents 0, 1, 2, 4, etc., and just add the cell names in the metadata. So some people do this. I sometimes do this. Sometimes I just rename the idents. I don't care and work with the data set right now and will not save it for the future work. However, it works like that. And now when we executed this rename idents, so the one line example is in the code I sent to you, which is yes right here. So rename idents 0 to my cool cells. You just delete my cool cells and write the true name. And then it will go a little bit back and plot the UMaps again. What we will find, but we'll group by. Yes, ident. And let's make a more precise look. So what we see now is nothing yet. Sure, it will be yes. So if we take a look at the right plot, because I turned the labels on and we see it, we can already see that you see the Kupffer cells, which you were identifying, the 3 cell markers, 3 cell clusters, are plotting very closely to each other and not that close to other cells. And do you remember we evaluated the T cell marker, which actually apparently really indicates the T cells somewhere in these clusters. So we have T cells 1, T cells 2, neutrophils, questionable, but totally can be still here. And B cells here. So probably these 3 clusters are of different B cell origin. For example, some of the plasmacytoid plasma cells, other cells are naive or memory B cells. These B cells are also required to be identified more detailed. But what is important here, we can see the separation of the cells which reside in the liver, so the liver macrophages which are always there, and the blood recruited cells, so these neutrophils. You can see that differently they cluster not together. They are of different markers according to this plot. Yeah, and in this case, so for example, you have this, what to do next, right? You have these 3 populations of the Kupffer cells. However, you would like to understand how are they different in the real life. And we know, I know, and I will share this knowledge with you, that there's one marker which we already evaluated, which indicates the quiescent or non-activated Kupffer cells. So we will just make it as a feature, and this marker is markable. So it's expressed in the non-activated Kupffer cells, but not expressed in activated cells, and it's very specific. And we make the feature plot. So just remember, the Kupffer cells are these bright buttons. And we make the feature plot for this only marker. And in this example, we see that among all the Kupffer cells we have, only these Kupffer cells, the lower clusters, 15 and this one, is markable, which means they're not activated and not taking an active part in the inflammatory process. Maybe they are tolerance cells. Maybe there are cells which are exhausted and cannot be participating in this information anymore. However, it's one of the ways how you can distinguish them. And how do you know these markers? You just search literature according to your data sets. You work with neuronal tissue. You have 10 clusters of neurons, you go to the literature and Google your markers. And that's how you can distinguish them further. Why is that important? Because if you can then compare how much these cells are different in the, actually, I will try right now to buy, I don't remember if it works or not, because what I would like to do to evaluate how these cells are different in different conditions. So I will try the group by. Okay, so we'll just do it like this. So we can see that in healthy, it seems that there is more cells of the inactive or the quiescent Kupffer cells as compared to the hepatocellular carcinoma. However, the hepatocellular carcinoma cells are mainly distributed here. So there is again a more precise way how to calculate exactly the number of the cells. Of course, it should be interpreted very carefully because you cannot be sure how in reality many cells are there because of sample preparation, because of the technical issues. However, if there is, for example, 0 cells of hepatocellular carcinoma in this cluster, you can make sure that something is different between these two conditions. That's very basic analysis you can make. However, it's not the most interesting thing, I guess. No, actually, so actually, I did the example for you how many cells per cluster can you find. So if you would just use a standard R function table, you can actually make this table based on the idents. So idents is the standard variable in the Seurat object, which shows this idents, which were named. So we will go to this object now. I think I can just view it. The second function. It will open, and we can see that we actually have a very small number of eta cells, which is good because eta cells are not immune cells. In the previous work with the data sets for you, I actually tried to filter out all of the non-immune cells. So it seems I just missed a little bit. However, we have many neutrophils and even more Kupffer cells, quite a lot, and yeah, and quite a lot of the cluster 1, which we still don't know what is that. But still, you can do the same with splitting by conditions. So we can just also compare how much of the cells are healthy versus diseased tissue. Yeah, if I will find some example, yeah, I will just show you how. But it's also again based on the standard. I just don't remember how to make it better. So probably it will not work because of unequal number. Okay, it's not exactly what they wanted, but do it in this way. So Kupffer cells, which are, let's say, Markov positive, which cluster? Okay. Nevertheless, in this way, if you would like to take a more precise look, you can compare how much of the Kupffer cells of this marker composition you had in one condition versus in another condition, and it seems that in healthy tissue, this S100A8 least plus cells quite a bigger number of those in the healthy versus hepatocellular carcinoma. And of course, you cannot say just from one data set that it seems the cells are not present in the diseased liver that much. However, if you compare a lot of data set, and we'll see that statistically significant, this number is different. And if we'll try to exclude all the technical variants, probably it's something more true than. However, there are of course more precise way to calculate the number of the cells, for example, flow cytometry. And actually, when you do the single cell RNA-seq analysis, you find the cell populations which were not known before, because it's not possible to distinguish one Kupffer cells from another, from the third one with histology, and there's 20,000 genes, and unlikely you just can randomly target with one marker and stain for it and distinguish it. So if you imagine that this is a new cell population never described before, you can now try to develop a protocol for flow cytometry sorting related to these markers, if it's possible. If there's superficial, is there antibodies, etc., and calculate more precisely how much of those. That would be one of the outputs of this analysis. However, what is actually interesting, and we unfortunately have not that much of time, so we can do the pathway analysis. the very basic one, and I will try to shrink the explanation in the 10 minutes, so we have a few more minutes to discuss. So we make the new variable my awesome cells, and actually we will, yes, so what I tried to do here, I tried to make you an example just based on the 2 cell populations, just because it will not take too much time or too much resources. So for example, let's compare the 2 cells. So we will take this very abundant cell population for healthy liver, copper cells as 188 positive versus which cells were also abundant. Actually, that's not that much of the cells. Okay, versus the same, but with S109 marker, just for fun. And let's take a look how different they are. So my awesome cells will use a Seurat function to subset from this big data set we have based on the idents or the cell cluster names which we assign. Copper cells of this marker and this marker. So we will just LDR do this one. Yes, R is happy. So and then we'll use a function which is the basis of the reactome pathway library, which will separately analyze the expression of these clusters for us. Usually it takes quite a lot of time, especially in big data sets. Considering that we just subsampled 2 cell populations, it shouldn't be that painful for us. So if we write calculating the average cluster expression, and in the meanwhile, I will take a look at the questions.
Alexander, I have a question.
Yes.
When you do a subset in the Seurat object, do you create a new Seurat object?
Yes. So
and you can save that one.
Yes, definitely. So, for example, if you would be a person studying only these cells and not caring about any else cells, you could do this. You could create your awesome cells object with subsetting the liver object with only B cells, and you could just collect 10 of those and then investigate them separately against each other. So it will finish the evaluation very soon. I see. Yes, it's already done, and we can actually take a look at this my awesome cells. So if we just print the name of the Seurat object, my awesome cells, it will show us that okay, there is a few assays, RNA and integrated, and there is 42,000 genes across 2,300 cells in this object. And if we compare it with the original object, we'll just write the name of it. Sorry, where it was my data. You can see there is a slightly more, so it's again 42,000 features, but again now across almost 12,000 samples. So we shrink it quite a lot just subsetting those cell populations. However, it's already finished with this analyzing of the expression because it makes separately for this package, and we can use a second function, which will go from the gene names to enrichment of the pathways. So it created a new object, pathway expression, and we can find it here, I guess. Yes, in the environment. Open that. So how it looks? We have 2 cell columns here. The score which was assigned to each pathway and the names of these pathways. So based on this score now, we can sort these pathways according to the maximum and minimum enriched pathways, I would say so. So first of all, and this is I just took from the very basic manual because you can obviously modify it and receive other outputs. We will just remove this Seurat. So we previously had it, it's a cell name, blah, 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 Seurat. Now we just don't have it. So it's a very supplementary thing. So now we select these columns and calculate the maximum minimal difference. And since we're already very close to the end, I will just execute it and we will discuss it already in the plot. So we'll make this very quickly. So the pathway with the biggest difference appeared to be the oleophenol metabolism, which was enriched negatively in the copper cells with S189 marker, and very positively you can see that the expression quite high in the copper cells is SA8. However, it's not the convenient.
What are the values of that score that it showed us? So what is the minimal value and what is the highest value, just to understand?
So there is no minimal and maximum because they're relative. And actually, the good idea would be to go for the pathways function. So what's the name of this package?
Okay.
So usually how it works, it just makes a specific score for the enrichment. Different packages use slightly different approaches for that. So no, actually we want the function description of the function. Maybe we'll find it right now.
Sasha, maybe I just understood Carolina's question differently. Like, what literally these values means? Did I understood it?
So
correct?
this is this course, and actually I found this description. Now I just need to find it.
Basically, what I wanted to understand is how do we decide that these are the highly enriched? Because I just saw 0.1 this, and
so basically
it seems low. I don't know what it is.
Yeah, so basically this number is a number. Well, so this algorithm scores all the pathways. So it takes the expression of the different genes, combines these genes in one pathway, and makes the scoring based on their own algorithm, which is possible to read in the function description. And this is the score. So if it's positive, it means that this pathway is expressed in the cell over enriched compared with other cells we compare these. So I don't see any really high numbers here, but we can sort for that, and so we can see the real low relative expression of the pathway is like minus 0.5, right, etc. And that's why we make.
I imagine that is a relative number relative to the rest of the pathways. And when you say that it is positive, is that is enriched with upregulated genes?
Yes.
Oh, okay.
That's exactly what it means, and that's why I very much like this heat map representation. So it's actually not very nice because we have only two cells, so and you cannot actually see all of the cell names. But when you add more clusters, it's actually more nice, or when you can just change some settings on this board. So here you can see the left is this S800A9 and right is S808 positive cells, which are very abundant in the healthy tissue. Here you can see the cover key of this enrichment score, which is relative enrichment score, and here you can see the most different pathways which were associated with these cells. So the next point would be here to understand whether these pathways are interesting for your specific condition or relevant. For example, this oleophenol metabolism is obviously very, very important for cells. So it's super enriched in the normal cells which are persistent in the healthy tissue and not that much in the hepatocellular carcinoma tissue. However, it's not for sure that this is a disease relevant pathway. So it's not like we can, target this pathway very easily just because of the knowledge of this pathway, or not likely it's super relevant for us in the context of separate markers. However, this is something already more interesting. For example, beta-klotho mediated ligand binding, and it appears very, very not. So it's minus 0.4, right? Not exactly enriched in the hepatocellular carcinoma cells. So for that, we can take this bold suggestion that maybe this specific metabolic pathway is affected somehow in the hepatocellular carcinoma. However, what we should really specifically consider is that we had a lot of less cells of this left identity as compared with the right identity. So the proper idea would be to make somehow equal representation of these both cells, for example, to get more data sets or to sequence more cells and then to compare them. Because for this left, we had like a few hundreds, I think, and for the right, we had like 1,500. So it can be one of the reasons like this technical issue. However, if we are very confident in our cell populations and we are sure that we make the quality control steps correctly, if we're sure that we made the integration correctly, and we can play around with this and see how it impacts our analysis, by the way, then we can actually make suggestions regarding the pathways which are involved or affected in specific diseases. And then I would imagine that we can take a look into specific markers in these pathways which are over or hyperexpressed, and then think about how therapeutically valuable they are. So read about them, see if there are any drugs targeting them, and make the next tertiary analysis. Because this is always a secondary analysis. So also in the standard rectum pathways manual, there was an example. You can check specific with pathways. If you remember, we put specific genes of interest over the cells, and we can do the same but with the pathways. Those are specific markers or pathways are for B cells, and what I would expect it will not give us a lot. It doesn't actually give us a lot because there are obviously not B cells. And I think I didn't actually add any legends here, so I cannot say which in which way it's over or hypo reached. But nevertheless, it shouldn't work like that because it's not Kupffer cells. However, if we would be really curious about it, we could find specifically something related to the Kupffer cells, or maybe power cytosis. So we see events related to the. Okay, this is quite specific thingy. Yeah, and actually they're not exactly that different in this specific case, because still they are both macrophages, but maybe and yeah, so the markers, the pathways I can think about are not actually that differently expressed in these cells. So either because we cannot calculate it adequately for the small cell population, or because they're so similar. Maybe it was in inflammasome already. How about I just cannot think about any good example to show you. What if we just take a look? Okay, so we can actually play around. So yeah, this one of the pathways actually detected related to the potassium channels is very differently expressed in these cells, and if we just change it all to one pathway, it should show us that yeah, it's differently expressed in this. I mean, it will be super ugly. Okay, it actually requires us two pathways at least, so I will add a random one. Make this. It will produce super ugly plot. It's just two markers, but yeah, that's the pathways. However, still, you see, from this example, you again cannot distinguish between these methods. It's not. Still, it's not the most optimal way either, because of the sample we use or because of the pathways. So I would say first to play around with the most differently expressed pathways, which we observe them, and then we decide if we would like to compare them with some other pathways. Of course, it's not a great example, again, because we selected quite a few cells for this analysis, and usually you select a few thousand of cells, or maybe tens of thousands of cells, and there it looks much more interesting. And even when you finish the workshop, you can just compare all the cells we detected and see how it's different. So just for fun, we can actually compare Kupffer cells with non-Kupffer cells. But just to make it look better, we will select the cells which is a good number. So T cells shouldn't be that similar, right? So cells is local. And it will take approximately 1 minute. So we'll try to reproduce this for cells. So calculating the expression should be done in like 30 seconds. So usually all this analysis is quite simply to perform on your personal laptops, because it's not resource heavy, and 8 GB of RAM usually enough for small data sets. However, if you work with a few dozens of thousands of cells, it usually requires you some access to the cluster, because it's really resource heavy at this point. Okay, so it goes very well. And there was also a discussion regarding the ranking. So again, it's not about the genes, but about the pathways in this case. And when it prepares the pathway ranking, the enrichment ranking, of course, it takes in accordance the expression of all the genes in the pathway. However, again, what's the name of this? It's gene set enrichment analysis. And actually, there's many ways how you make it. And the Reactome pathways is just one of the specific examples referring to one specific database, which is named Reactome. Type Reactome database. Yeah, you can actually go there even without any R and check the pathways you're interested in. However, the analysis is over, so we can take a look. And here, it's already something different. So we see the different pathways, and still somehow some of them are actually similar expressed here. However, you see they're totally different as compared with the previous comparison of the only capture cells. So, for example, you see that what would be a good example here? Again, oleophenol metabolism is super expressed in capture cells, as we already know from the previous analysis, and it's not exactly expressed in the T cells. Yeah. And you can play with that around again if your sample allows. So I think it would be the end of the practical part, and I think I will still have like 10-ish minutes for the discussion, so we can refer to some of the details. However, I would also love to give you some homework. So if you feel like that, or you just want to do this, you can do this, and we can discuss your analysis. Now I am copy pasting the homework. Okay, so I will send it to the channel day number 4. Okay, so the first task would be to find healthy versus treated data set. So you can either use this data set, or instead of treated, I mean the conditions. You can either find healthy versus disease treated, or just disease, or you can just use the same data set we just worked with. Perform the clustering and import the identification of the clusters as we did, but you are free to actually do the whole identification. So usually when you know the data set, or the cell markers, you can do it quite fast. So it's below 1 hour if you just see the markers, you're sure in the most of those. But I would say it could take around 1 hour with the database, and also try to to distinguish them as precisely as possible. Like if you see different T cells markers, just try your best guess and identify which exactly these T cells, either memory cells or something, may also require you to search the literature. Then make the pathway analysis and interpretation, so you will have more time at home. So maybe it's also nice in this case, for example, to Google about this pathway in the context of the hepatocellular carcinoma. Maybe it's already known. And then number 4 and number 5 is actually about the gene ontology analysis, which we don't have time to do today. However, the code is present here. So it's a little bit different way, which we discussed a few days ago, to make some biological meaning from your genes. So it again performs enrichment and interpretation of the gene sets and according to the gene ontology database records. So you will see something similar to that in the output. However, it would be a homework to do it independently and ask if something doesn't. So with this, I'm happy to stop sharing the screen and to ask about your opinions / results / it sucked / it was great, and any further comments you would like to add.